Der Sonderforschungsbereich 599 der Deutschen Forschungsgemeinschaft zu permanenten und resorbierbaren Implantaten beschäftigt sich mit einem wichtigen Problem der heutigen Medizin. Bedingt durch zunehmende Verschleißerscheinungen der Gelenke, aber auch Sinnesbehinderungen, steigt der Bedarf an Implantaten, um diese Funktionsstörungen wieder zu kompensieren. Ziel ist es, Implantate zu entwickeln, die ihre Funktion optimal erfüllen, gleichzeitig aber auch die Reaktion mit dem Körpergewebe so gestalten lassen, dass es zu den gewünschten Effekten kommt. Neben den Implantaten, die dauerhaft im Körper verbleiben, wie zum Beispiel Gelenkprothesen oder sogenannten Cochlea-Implantaten zum Ersatz des ausgefallenen Innenohrs, gibt es Implantate, die nur vorübergehend im Körper benötigt werden. Dazu zählen vor allem sogenannte Osteosynthesematerialien, also Werkstoffe, die bei Knochenbrüchen oder bei Verlusten des Knochen eingesetzt werden. Der Sonderforschungsbereich versucht, diese Ziele zu erreichen durch eine besondere Form der transdisziplinären Zusammenarbeit, die sich nicht nur auf verschiedene Fachgebiete, die für diese Implantatentwicklung erforderlich sind, erstreckt, sondern auch verschiedene getrennte Institutionen am Standort Hannover und Braunschweig zusammenführt. So ist es in jedem Projekt garantiert, dass die jeweils erforderlichen Experten aus der Universität Hannover, der Tierärztlichen Hochschule, der Medizinischen Hochschule und dem Laserzentrum zusammenarbeiten. Unterstützt wird die Expertise durch das Helmholtz-Zentrum für Infektionsforschung in Braunschweig und durch die Technische Universität in Braunschweig. Von besonderer Bedeutung ist dabei die Kooperation zwischen den Ingenieurwissenschaften, den Naturwissenschaften und der Medizin. Die von der medizinischen Anwendung her definierten Probleme und Fragestellungen werden in einen Entwicklungsprozess eingeschleust, der von Ingenieuren geleitet, durch die Naturwissenschaften unterstützt und begleitet wird. Die dann erzielten Ergebnisse müssen sich anschließend in einer biologischen Überprüfung bewähren, sodass sie dann tatsächlich wieder im Hinblick auf die Verbesserung der medizinischen Ausgangslage getestet werden können. Dank der Förderung durch die Deutsche Forschungsgemeinschaft ist diese Form der Grundlagenforschung mit Orientierung auf die spätere Anwendung möglich. Bevor jedoch eine angewandte Forschung und die Überführung in die Translation hin zu definitiven Produkten erfolgen kann, muss diese Grundlagenforschung wertfrei und auch unabhängig von späteren Verwertungsabsichten erfolgen. Gemeinsame Studieneinrichtungen wie Masterstudiengang Biomedizintechnik und bestehende PhD-Programme runden die Nachwuchsförderung ab.
Around 15 million people in Germany are affected by hearing loss. Often, the cause is damage to the inner ear, also known as the cochlea. One means of helping patients with inner ear deafness is the cochlear implant, or CI. The sensory cells in the ear are called the inner hair cells. After they die, the nerve cells or spiral ganglion cells to which they are connected also degenerate. Sounds are transmitted via a microphone to the implant from where, in the form of signals, they are passed on to the brain via electrode contacts. As present-day implants have only limited capacity for signal transmission, hearing with the cochlear implant is not the same as natural hearing. The researchers at Collaborative Research Centre SFB 599 are seeking to close this gap. This will, for example, mean increasing the number of channels or electrode contacts. In order to be able to exploit these improvements, the electrode nerve interface needs to be optimized, thus also enhancing the transmission of the auditory signals. One special focus here is on reducing the growth of connective tissue around the electrode array. Using an atomic force microscope, the growth of a connective tissue cell can be assessed, for example, and the adhesive forces holding it in place can be measured. The aim is, by modifying the surface, to prevent this growth in the future and thus optimize the implant's efficiency. Another priority is improving the survival of the nerve cells that require stimulation. There may even be potential here to form new cells. To achieve this, the electrode surface needs to be altered. Among the aims of the current lines of research are the creation of different structures, coatings or proteins, and the use of specially modified cells. Every year, around 1 million dental implants are placed in Germany alone. Unfortunately, however, not all of these implants last as long as desired or envisaged. There are a number of possible reasons for this. One is a change in the surrounding tissue, resulting from inflammation caused by bacterial buildup, which forms a biofilm known as plaque. In order to obtain good long-term outcomes for dental implants, the implant materials and surfaces must be designed to ensure the body's own tissues adhere well to the implant and must have adequate resistance to chemical and physical stresses in the oral cavity. To reduce plaque deposits and to lower the susceptibility of ceramic implants to fracturing, different ceramic materials are first tested as to their corrosion properties. All developments in this field are tested in long-term biomechanical stress experiments using a chewing simulator. Structural changes to the surface can then be viewed microscopically right down to the last detail. To enhance their understanding of the complex interactions between implants and tissues, the researchers are seeking to characterize the way in which the gums interact with the implant surface and with the biofilms that form on them. The methods used provide useful findings, including information about the exact bacterial composition of the biofilms and their three-dimensional structure. The aim is that, based on these findings, relevant implant surfaces and biological factors will be optimally combined. The surrounding mucous membranes of the mouth will thus adhere better to the implant, while formation of biofilm is reduced. Cardiovascular conditions are among the commonest causes of poor health and mortality. One aim in the surgical therapy of these diseases and disorders is to replace damaged heart and vascular tissue by means of biological prostheses. In Collaborative Research Centre SFB 599, stomach tissue taken from the patient is stabilized using a resorbable supporting mesh made of magnesium and then implanted. This ensures, during the healing process, that the prosthesis is sufficiently stable even in the high pressure area of the heart and the aorta. Over a predefined period, the magnesium mesh is broken down piece by piece. What remains should be a replacement tissue that is hardly distinguishable from the original healthy tissue. The alloying elements are melted in a crucible at about 750 degrees Celsius. The moulding process requires a lot of experience, as the filled crucible needs to be emptied fully and evenly into the mould. Using an extrusion press, the magnesium alloys are formed into shapes. This involves the starting material first being preheated in a convection oven and then formed into the shape desired using the force applied in the press. Abrasive water jet technology involves a strong jet of water being generated under high pressure by a fine nozzle. Abrasive sand particles are added to the water jet and these end up striking the workpiece. This enables almost any shapes to be created from sheets of material. The magnesium meshes are tested using a dynamic test facility to see what load change can be achieved. The membrane moves in a similar way to the heart. The corrosion can be tracked by video camera up to the point where the mesh breaks. Initial clinical applications involving the right atrium, which is the low pressure area of the heart, have already been successful. The latest promising research outcomes give hope that it will soon be possible to use this innovative therapy option in the high pressure area as well. Human joints, in the knee for example, can be seriously damaged by an accident or through wear and tear, as in osteoarthritis. Every year in Germany, more than 100,000 patients, some of them young, are fitted with an artificial joint made of metal and plastic components called an endoprosthesis. The aim of an operation of this kind is to allow pain-free mobility, providing greater independence and enhancing the quality of life. To increase the durability of a replacement joint and to prevent allergic reactions to the metal, which occur only rarely, the researchers involved devote particular attention to developing new implant materials. Collaborative Research Centre SFB 599 conducts fundamental research. Among its objectives are the production and processing of implant materials and the functionalization, calculation-based design and testing of implants with high rigidity and durability, with a view to preventing the need for surgery to replace them and the accompanying requirement for rehabilitation. In the Biomechanics and Biomaterials Laboratory, Researchers simulate the way the knee joint moves and the conditions to which it is subjected. A prosthesis secured in this testing instrument must be able to withstand many millions of movement cycles in order to prove its future suitability for use in the body. When developing new materials, it's often worthwhile taking a look at the optimization strategies found in nature. For example, the hard mother of pearl found in the shells of pearl oysters is being investigated as a natural model that may inspire the design of break-resistant components. This is because it possesses ideal natural structures, which serve as components for a prosthesis to replace the knee joint. In order to determine how robust a material is, it is first placed in a test rig. With both rolling and sliding movements, the stresses on a human knee are simulated and then analysed. Developing and producing an artificial material with highly similar properties to mother of pearl is one of the stated aims of the SFB 599 project. Implant materials used with bone, and especially those used with joints, need to be highly stable because of the loading they are subjected to. However, to avoid stress peaks and resulting fractures, they need to be sufficiently flexible as well. Because they are very hard, ceramic materials are already in use as implant materials, even though their flexibility is very low. As so often, we find models in nature in which the advantages of hard ceramics are combined with a flexible polymer material to form a composite. Mother of pearl, which is found in seashells and snail shells, is just such a composite material. Thanks to its well-structured design and its combination of hard ceramic platelets with a biopolymer, the fracture toughness of a seashell is 3,000 times as high as that of the ceramic components alone. In Collaborative Research Centre SFB 599, the design structure of mother of pearl is being replicated in a multi-step process. The particles generated by this work are platelet-shaped and only a few micrometers in size, which is why they're not visible to the naked eye. A special robot, called a dip coat robot, dips a substrate first into a suspension of the created particles and then into a polymer solution. In this way, layer by layer, a structure is created which resembles that of natural mother of pearl. A second method for creating mother of pearl-like structures is directional solidification. When freezing a blend of polymers and ceramic particles, the particles align themselves. If the water is then removed from the specimens, this leaves behind structures that are similar to mother of pearl. It's necessary to investigate those properties of natural mother of pearl and of artificial mother of pearl-like materials, which are important with regard to their workability. Various methods, such as scratch tests, are used in order to explore how these composites behave in terms of material separation. It's also important to investigate the wear behavior of these materials under stress. Combined rolling sliding experiments are performed to simulate the stresses which the knee joint is subjected to. Two fundamentally different types of materials are used in medicine for the treatment of fractures. Non-resorbable implants, made of titanium or steel, are utilized primarily for fractures in weight-bearing bones and generally have to be removed in a second operation after the break has healed. Resorbable implants, derived from certain lactic acids, are also used. As they are less stable, they are used in surgery of non-weight-bearing parts of the body, such as the hands and face. The researchers in Collaborative Research Centre SFB 599 are looking for new resorbable materials that have sufficient primary stability to enable them to be used even with weight-bearing bones, and that are broken down by the body once the fracture is healed. In initial tests, magnesium-based materials showed promising results. When developing different magnesium alloys, various properties are crucial in selecting the individual alloying components. These properties include how well tolerated the various constituents are, as well as mechanical stability and workability. After moulding, the material is worked to form smaller specimen geometries using a large extrusion press. Specific modification of the surface enables the roughness and peripheral zone properties to be altered, so that the degradation or stability of the implants can be influenced. Detailed cell culture investigations are being followed by studies on the intact whole body. Equipment is available for assessing the breakdown of the implant in the bones and how well tolerated the implants are. This includes a modern microcomputed tomograph. The important assessment of how well the body tolerates these magnesium implants takes place afterwards in the form of histological studies on the bone implant composite and the surrounding tissue. Here, state-of-the-art microscope technology offers a wide range of options for analysis. After all the findings have been collated and evaluated, the ultimate aim is that a new, optimal magnesium alloy will be available as an implant material for use with weight-bearing bones. Intelligent osteosynthesis, in other words. This will make a second operation to remove the implant unnecessary.